first week is morula blastocyst. Blastocyst started implantation. That is first week we saw. Second week, 2 layers, week of 2. So 2 layers, epiblast, hypoblast, syncytial trophoblast, cytotrophoblast, 2 cavities, amniotic cavity and yolk sac. And lastly, chorionic cavity or extra embryonic coelom also formed in the second week. Third week, we gastrulation, that is a very important change in the third week. 3 layers are formed. What are the 3 layers? Ectoderm, endoderm and intraembryonic mesoderm. Primitive streak is formed in the third week. Notocard is formed in the third week. So from third week is the embryonic period. We have started the embryonic period in the last class. Now we will continue the fourth week. In this, the general embryology will be over, except the placenta. So the fourth week is the busy week. Busy week. Even though I will say main events in this fourth week like neurulation, somites, foldings, but mainly I will concentrate on the neurulation and somites. Foldings of the embryo I will tell very briefly, but you have to understand that this is embryonic period. Embryonic period is from third week to the 8th week. So in this embryonic period, all the organs are formed. So we are not going to tell all the organ formation, the heartbeat, starts, that all again we will say separately in the cardiovascular system development. So leaving that organ formation, limb buds start developing in the fourth week. Fourth week to 8th week, as I said, whatever embryology question is asked, fourth week to 8th week is the period where all the organs are formed. Liver development, pancreas. So we have to remember simply we have to write only fourth week to 8th week. So we are doing now fourth week in this class, but we are not telling all those events in this class. Leaving those that will be covered in the systemic embryology. Mainly what are the other changes in this we will see is neurulation and the somites. Little details about the foldings of the embryo. So this is first I will start with the chapter 1, neurulation in motion. That is the first chapter in the fourth week, neurulation. So you can see in the earliest days when the ectoderm is lying, so in the ectoderm you can see when it is only here, the ectoderm, now it is going to form the brain and spinal cord. So you have to remember ectoderm is the derivatives of ectoderm. If it is asked, you have to remember the nervous system is formed from the ectoderm. Nervous system. That part of the ectoderm which gives rise to the nervous system is called as neurectoderm. Ectoderm, now it is called as neurectoderm. It is working under the influence of the notocard, secondary organizer. Notocard is giving, it is lying deep to the ectoderm. It is giving signal to the ectoderm so that part of the ectoderm is now changing to neurectoderm. That will give rise to the future brain and spinal cord. Otherwise, central nervous system is developing from neurectoderm. The peripheral nervous system, again I will say next. So that is the beginning of the neurulation. Which is going to give rise to now the central nervous system? Neurectoderm. You please remember, ectoderm is going to give rise to the central nervous system. So this neurectoderm, when we say this third to 8th week, we can say in three parts. pre-somoid period, somoid period and post-somoid period. What is somoid period? Is 4th week. Somoid period, even this, do you know the size of this? When we are saying so many changes, do you know the size of this embryo? It's in few millimeters. So to understand the size of this or the gestational age of this, we see under 3 parts: pre-somoid period, somoid period and the post-somoid period. What is the somoid period? 4th week. So this is what we said. It is new ectoderm, which is giving the signal to this ectoderm to form the central nervous system is notochord. So you have to know this. Notochord is the secondary organizer. So you can see in this picture. So this is the three layer germ disc. So this is the notochord. Notochord is giving signal to the surface ectoderm. So that part is now called as the neural plate. So this is now neural plate. Notochord stimulates the development of the neural. First it is forming neural plate, but it is growing little more. So this neural plate will now grow like a groove, neural groove. And the raised margins will be called as neural folds. So that is the next development. What is the first stage of development of the neurulation? Neural plate. You can put like a flow chart. Neural plate. Next is neural groove. You can see in this picture which is neural groove. It is given. It is understood. So this is the neural groove. And this raised margins will be called as neural folds. The lateral margins, raised margins are called as neural folds, and the depression is called as neural groove. So that is the next stages. You can write neural plate, arrow. You can put neural groove, neural folds. So this is what we said. Groove is called as neural groove, and raised margins are called as neural folds. Now it will form the neural tube. One groove is there, folds are there. To form neural tube, it has to fuse. Neural folds have to fuse, and it has to bury into it has to detach itself from the ectoderm, so that it will go under the ectoderm. That will be the formation of the neural tube. That is the next stage. Neural groove, neural folds now has to fuse. So before that I have to say one more point. Neural crust cells. You please note of this. This is one important part of this development. Neural crust cells. Maybe ask us short notes for you. So the raised margins are having specialized cells. Neural crust cells. Even some embryologists believe that it is the 4th germ layer. That much importance is this neural crust cells. If I say its derivatives, you will be wondering, it will form in the skin melanocytes. It will go to the tooth and form the odontoblast. It will form the facial skeleton and muscles. Then it will form the peripheral nervous system. What is peripheral nervous system means what? Ganglia, spinal ganglia. It will form the autonomic nervous system. What is it? Sympathetic chain, sympathetic ganglia, parasympathetic ganglia. It will form sympathetic and parasympathetic ganglia in the head and neck region also. And you see its derivatives are vast. It doesn't come under any group. It is forming one side cells, one side system, one side skeleton. So that's why some embryologists call this as the neural 4th germ layer. So you have to appreciate its importance of this neural crust cells. So the tips of the neural folds are having so we have to think that these tips are having specialized cells, that is called as the neural crest cells. So now next is the raised edges are now going to close, so that like zipping, these two edges are going to close and it is going to complete the neural tube formation. When this process is taking place, you have to think in which order it is closing. Whether it is adding closing from cephalic to caudal or caudal to cephalic, it is not in both the ways. It is different. It starts in the cervical region first, then extends above and below. This neural tube closure first starts in the cervical region, then it is closing above and below, leaving two openings in the upper part and the lower part, that is anterior neuropore and posterior neuropore. Actually, it should be named as cranial neuropore and caudal neuropore, but the names are anterior neuropore and posterior neuropore. So that is the neural tube formation. So this may be, the question may be neurulation. Then you have to start from neural plate, neural groove, neural folds, then this closure, then that process is called as the neurulation. So as I said, it starts first in the cervical region, then it extends to the above and below, leaving two openings. That openings are called as anterior neuropore and posterior neuropore. So this is one MCQ in the PG entrance. Anterior neuropore closes at 20 somite stage and posterior neuropore closes at 25. As I said, pre-somite period, somite period. In the somite period, we used to refer the number of somites present in the embryo. So how many somites are formed? So that is 20 somite period, that is anterior neuropore closure, and posterior neuropore closure is in the 25 somite stage. So if they don't close, what will happen? I said simply, very easily, it is closing, then leaving two openings. This opening is also closing, I said. You have to now think if they don't close. Yes, they don't close. In some cases, they are called as neural tube defects. Neural tube defects. So they are very common, but nowadays it is reduced with the gestational period. Folic acid is mandatory prescription. With this, they have understood this neural tube defects have been reduced. What are the neural tube defects? So we will see before that these images. So this is what we said. The anterior neuropore is, now you understood, this is the closure in the, first in the cervical region, then it extends above and below. In this you can see the tips of the neural folds are having the specialized cells, neural crest cells. So this is that, like that you have to draw the diagram. A neural fold, you have to put another color in the edges. That you have to name it as neural crest cells. So the folds meet and form the caudal closed part, that is called spinal cord. Cranial is having the hollow brain vesicles, forebrain, midbrain, hindbrain, so that the brain formation is complete. This neural tube appearance is retained in the adult in the spinal cord. Spinal cord you can appreciate this tube-like structure. But this brain development, again we have to tell. If there is time, we will tell in the last. Otherwise, you have to learn. It is having this tube-like appearance. How it is going to form this forebrain, midbrain, and hindbrain, and lastly this adult brain appearance. So we have completed the neurulation. So now I will add this little of this neural crest you are seeing. Neural crest, now you can see it has buried below the ectoderm. How it starts moving. it starts moving to various parts. This neural crest cell migration is very important. Without that, even phase will not develop normally. Treacher-Collins syndrome, if you search in the web page, Treacher-Collins syndrome, the face will be asymmetrical. So Treacher-Collins syndrome, they say this neural crest abnormality. So many abnormalities will happen if this is simply a very less emphasized in the embryology class, but you will appreciate its defects in the clinical side. So the neural crest cells migrate away vastly. They form one place nervous system, one place they form the cells, one place they form the phase. The major part of the human phase development is influenced by this neural crest cells. So this is a very interesting research topic, neural crest cells. So this is for neural closure, neural tube defects. The tube is not going to close completely. So if the tube is not going to complete this, we saw in the beginning notochord influences the development of the nervous system. If this nervous system closes only, axial skeleton will come and close. Vertebra will come and close this. If this neural tube is not closing, then the vertebra is also open. So that's why it is called as spina bifida. So only vertebra is open, then some part of meninges are coming out, some part of spinal cord is coming out. So they are the various types of spina bifida. So this is another malformation, that is anterior neurophore is not closing. So the cranial part of the neural tube, this anencephaly will not survive because the brain development is affected. So this is the anterior neurophore closure defects. The other one that is the caudal part closure. So this is what even the spina bifida occulta means no symptom is there. Meningocele is meninges are coming out, only meninges, no nervous tissue. If nervous tissue is coming out, meningomyelocele. So this completes the neurulation. So that is the chapter 1 in the 4th week is neurulation. You can note down this. I have given the surface ectoderm derivatives. You can note down this. As I said, nervous system is included in it is even given neural crest also. You note down this because that may be asked as short notes. You can see this neural crest, peripheral nervous system. As I said, Schwann cells, neuroglial cells, sympathetic nervous system, parasympathetic nervous system. In this, we have to add spinal ganglia. It is not in that given. Spinal ganglia also you add, peripheral nervous system. Apart from that, adrenal medulla, melanocytes, you note that adrenal cortex is some other derivative. Adrenal medulla is from neural crest. Again, this adrenal gland is an important research topic. Cortex from some other source, medulla from some other source, both are having different functions. Both are embryologically different, structurally different, and functionally also different. That is the very interesting adrenal gland. You can see this neural crest in the dermis of the skin give rise to the melanocytes. As I said, it gives the facial cartilage. Without that, our face cannot develop properly. In the tooth, you can see dentine, which is secreted by the odontoblast. So all these are the derivatives of the neural crest. And interestingly, you should see the neural tube last point, retina, and one more is posterior pituitary. Posterior pituitary. These are all derived from this neural tube only. Posterior pituitary. Again, pituitary gland development is an interesting topic. Anterior pituitary from some other source. Posterior pituitary directly from the nervous system. That's what it will have the nerve tracts in the posterior pituitary. And this eye development is again a very interesting topic, and it is a very composite topic that will lead to so many abnormalities in the eye development. Each part will be coming from some layers. You can see retina is directly from the nervous system. Retina. So that is what it is added. You can see in the ectoderm derivative, lens and cornea, as I was saying, this eye development is again composite. Lens and cornea from surface ectoderm, retina from neural tube. So like this, you can see in the ectoderm it is given anterior pituitary, posterior pituitary is from neural tube. So all this, if you at least note this, if we are asked pituitary, all this you can note. So this embryonic period, differentiation of 3 germ layers. One germ layer I have given, ectoderm, and we have covered the very important process, neurulation. So the next chapter, as I said, folding, which we will not see in detail in this, because if I have to say folding itself is a one class. Totally I have, if I want to tell you very detailed. So we are not telling in the detail in this class. We will tell what are the folds. What are the names of the folds, we will see. So the second chapter in the 4th week is great folding begins. So great folding, as I said in the last class, still now the embryo is flat. So now you have to, if you are taking one piece of paper, it is like piece of paper. 3 pieces of paper. It is not having any shape, any form. So that is the trilaminar germ disc. In the 4th week, so this germ disc bends cranially. You have to bend the paper cranially. Bend that again caudally. They are called as head fold and tail fold. Again you have to bend this laterally also. Bend that paper laterally. So that what is that you are having a cylindrical embryo. Again, if we say very simple like this, you imagine if this folding is not proper, if they don't close. If they don't close, what will happen? Again, many defects in the anterior abdominal wall or in the thoracic wall, lateral foldings, if they don't close, heart may be outside. Have you seen? Ectopia cordis. Ectopia cordis may be outside. You can see the intestines hanging outside, omphalocele. So all these are the defects of these foldings of the embryo. These foldings, if they are not doing appropriately, then we are not going to get a closed body. So this folding is very important. We will see only the names of the folds. So now you have to, as we started in the trilaminar germ disc, it is not having any shape, 3 layered, simple layered trilaminar, ectoderm, endoderm and mesoderm. So now what causes this folding is neurulation. The neurulation is occurring in the middle. So the embryo is growing in the middle. That causes the head fold and tail fold. The somites are developing on the lateral side. That causes the lateral foldings. So the expansion of the embryo in the midline and also on the lateral side. So at least note the names of the folds. Head fold, tail fold and lateral folds that starts at the end of the 3rd week or the beginning of the 4th week. What is the major change in this folds? One, as I said already, what is that? shape of the embryo. So that is one major change in this folding. There are many changes we have to see, like changes in the situation of pericardial cavities, changes all that. But major change is change in the shape of the embryo. So this is what it is growing so fast, so that now head fold, tail fold and lateral folds, if they are not folding, so otherwise we can't get the closed body wall. So this is that we have to see in this. So if you see this first picture, that is the trilaminar germ disc. So this is like ectoderm, mesoderm,